tandem repeats, okay, to develop your unique fingerprint, okay? And so these are just regions of DNA that contain short, tandemly repeated nucleotides, right, ranging anywhere from 2 to 6 bases, okay? And this number of repeats, they vary frequently between individuals, and they're classified based on the length of that piece, right? So if you have a repetitive sequence of 2 nucleotides, is it a dinucleotide 5, so on and so forth, okay? The short tandem repeats are widely found in both prokaryotes and eukaryotes, including humans, which is why we can take advantage of them to develop our fingerprints. And they only account for about 1 to 3% of the human genome. I think this number is actually kind of changing. The most recent reference I have is from 2020, but I do think that they have since kind of updated it. They're talking about a little bit more than that. Most of them, not all, but most of them are found in non-coding regions, okay? So only about 8% are actually found in coding regions of the genome. And these are typically passed from parent to offspring without any change to them, okay? And so just, a little bit of refresher in terms of heredity, okay? So, when we're talking about a genetic locus, we're referring to the location of a gene or DNA sequence on a particular chromosome. And at each one of these genetic loci, right, you have what's known as alleles, okay? These are alternate forms of DNA at a given locus. And so for any type of gene, right, you can either be homozygous, meaning that you have identical alleles at that genetic locus. So you've inherited the same allele from both mom and dad, you're homozygous, or you can be heterozygous, meaning that you have different alleles at that given locus, right? So you've inherited a different allele from mom and a different allele from dad, okay? So when we're thinking about these short tandem repeats, okay, we're thinking of them as being at a particular genetic locus, right? You're going to inherit one copy from mom and one copy from dad. It's going to have a particular sequence to it, right? So this one happens to be a tetranucleotide, because it has 4, okay? And it's going to have variable amounts of repeats to that, okay? So in this example, you've inherited a version where it has 14 repeats from mom and 12 repeats from dad, okay? So if we want to characterize what that means at that loci, we would say that this individual is heterozygous for this particular STR, okay? And we would call their alleles as 12 and 14, okay? If we look at another one, right, we still have a tetranucleotide. This time, this is the particular sequence to it. You've inherited 10 repeats from dad and 10 repeats from mom. And so then in this instance, this individual is homozygous 10-10 at this particular short tandem repeat. Does that make sense so far? So, just as a refresher about how we go about getting your sample and how we run these samples, right? So I forget which night, maybe night 2, 3, and they actually swap. No one remembers, right? It feels like forever ago. Regardless of what night it was, right, we gave you some loops and you isolated some cheek cells, helix, K, incubation step, and you basically got your genomic DNA from your cheek cells, right? And then we used a Promega profiler kit, okay? And all this kit has, and this is a simplified version, is it has a bunch of different primer sets for each short tandem repeat that we want to amplify, right? So this is simplified, it only has 4. The profiler kit we use has, in fact, 10 STRs, so really there's 10 different primer sets. And this is actually quite a small profiler kit. And so Steve and I were just talking about this yesterday. I think the FBI, so their coded system, actually they've since introduced it to 20 different short tandem repeats. And we'll kind of get into why that is a little bit later. Right? But so basically we do this multiplex PCR reaction, and then we submitted it for sequencing on our 3500 genetic analyzer. Okay? And so how this works is that each one of those primer sets, okay, has some sort of fluorescent label on it, okay? So when we're looking at this chart, all the way to the left, these are the particular short tandem repeats that we amplify, okay? And so those are just their names to it. The next column is showing you what that fluorescent tag is, and so why we're able to detect it, okay? So there's 3 different channels in this particular profiler kit, and they all fluoresce at different wavelengths, which is why we're able to detect each one individually. And so I have them, they're listed in this column and also down below. I do just want to point out that when we do look at your chromatograms, okay, anything that has this TMR is actually, it's not going to be yellow on your chromatograph, and it's kind of hard to see. So your peaks are in fact going to be in black on your chromatograms. But you'll notice that the other 2 are in fact green and blue, okay? But I just wanted to point out that they're black on these chromatograms, okay? And they've listed, you know, basically what that particular STR is. And you know, if you want more information about what these STRs are, you can look those up, and their location on chromosomes, right? So the genetic locus. Right, and so this is, again, this is the actual profiler kit that we use. So again, this is just kind of a different view of the exact same table I just showed you. Okay, so you have 3 different windows because there's 3 different fluorescent tags, okay? And within each one, we've listed this particular short tandem repeat, okay? And so these, again, are the particular fluorescent tags that get added during the PCR amplification step, okay? Okay, now this looks kind of crazy, okay? But in addition to running your samples on the sequencer, okay, there's 2 other things that always get run at the same time. One is basically like a molecular weight marker. It kind of just helps, you know, it's because every run is going to run a little bit different, and so that's basically there just to help, what's the word I'm looking for? It's a reference, yeah, so things are going to migrate for each run, right? So just like any other type of, you know, PCR reaction, you always run a molecular weight marker with it. So that's always run too, it's an internal molecular weight marker. And then we have what's known as the allelic ladder, okay? So again, this is like shown you on the previous 2 slides, we have 3 different windows corresponding to the 3 different fluorescent tags, okay? And then the reason why there's so many different peaks is it's basically showing where you could expect any one of the allelic versions of that short tandem repeat to show up, right? So, you know, this is DWA STR, you could have 10 repeats all the way up to 22 repeats, right? So it's just showing you, this is an allelic ladder where you can expect to see each individual allele for a given short tandem repeat. Like when they're not whole numbers, it's because some of them have a portion of the nucleotide in it, right? And some of them.
So it's like 11.2, then 11 repeats, and then 2 more nucleotides.
So not a full.
So the point 2 is not really a fraction. The point 2 is telling you how many nucleotides of the next repeats are.
What is X and Y? Our peaks have that too.
Yeah, so amelogenin is basically looking at the sex chromosomes. So, yeah. Any other questions? No? Okay. Okay, so how do we actually go about reading your chromatogram, right? So hopefully everyone should have one at this point, right? And everyone should pretty much look like this, okay? And so how do we actually go about reading it? The first thing you're going to notice, okay, in gray boxes above the peaks is going to be the actual short tandem repeat locus, right? So it's going to be one of those 10 that I had listed out on the table. Here I have TH01 circles, so that's just one particular short tandem repeat, okay? And you'll notice that there's gray boxes above all the peaks that correspond to the particular STR. The next thing is that number underneath the peak. okay? That's basically your allele, right? It's telling you how many repeats of that sequence you have, okay? Now again, we kind of went over this, but we can be homozygous or we can be heterozygous for a particular STR. The way that we know what we are is you'll notice if you only have a single peak for a given STR, it's because you're homozygous for that particular allele at that STR, right? So this individual is homozygous 1313 for the D5 STR. Does that make sense? At the same time, you can also be heterozygous, right? So this individual is heterozygous 1112 for the D7 allele. You good with that? Yes.
What is the meaning of the type of?
So it's measuring how much fluorescence is detected, so that's usually indicative of how much PCI amplification. For our purposes, it doesn't matter, it's going to make the call if it detects a peak, but yes, usually the higher the peak, the more fluorescence, the more amplification there was. The problem does come in if you have very little amplification, sometimes it can call something wrong, or it won't call it at all because it's not actually detecting it as a peak, but that's where the molecular weight internal standards and the allelic ladder come into play for that. Yes.
I don't know. The reason you have the different P types has to do, as Allison said, with the efficiency of PCR. And remember, there are 10 PCR reactions going on in this tube simultaneously. You should not be surprised that some of those PCR reactions are more efficient than others, depending on how well the primers bind to the template.
But the peak doesn't have to do with the number of peaks. So you'll see like for 11, 12, those are different peaks. So it's not like the higher your peak, the more repeats you have. That's not what it is.
Right, it's only corresponding to the fluorescence, which corresponds to PCR efficiency. Make sense? Alright, so why do we use STR profiling, right? So one huge application is, I don't know if anyone does tissue culturing, cell culturing, right? Cell line authentication. This is can be a big problem in research, right? You're carrying out all these experiments on cell lines, they're behaving in certain manners, and then come to find out they're not actually what you thought they were, right? So this little cartoon is referring to a point in time when, so why is this kind of a funny joke? Right, have we all heard of Gila and Henrietta Lacks?
Yeah.
Okay, so this is a cell line that was acquired from a black, genetic female, okay, back in, the 50s, who had cancer, okay, these cells were taken without her consent. If you haven't read Rebecca Sloot's, story about this, it really shows the immortal life of Henrietta Lacks is very good. But the reason why this is a cartoon, right, is because again, genetically female should have no Y chromosomes. HeLa cells, when they were first ramping up production of them, there was a big issue, and they were contaminating labs everywhere. Right? I mean, it was amazing because these were the first immortal cell lines that were, produced and sold commercially and whatnot. But it did end up turning into a very big problem because labs everywhere had HeLa cell contamination. Okay, so you were doing these experiments thinking you were doing it on one type of cell line, only to come to find out we're actually doing it on HeLa cells because you had contamination. Okay, so a lot of times now, a lot of journals will require you to do STR profiling to authenticate the cell lines that you're working with before you can publish in your journal. Determining twin zygosity, right? Why do we think that STR profiling is useful for determining twin zygosity? And does everyone know what I mean when I say twin zygosity? So whether you're identical or fraternal? So why is this useful? How would we know? Right? So identical twins will have exact same fingerprinting, right? Fraternal twins will not. Forensic applications, right? Paternity testing, helping to identify missing persons, and then of course in our instance, right? identifying perpetrator. Committed a heinous crime. Heinous crime. Right, I didn't really need a picture, we're present for it. You see what was done, right? So what we're gonna use SDR profiling is to find out who did it. Okay, so how are we gonna do this? Well, we collected evidence, right? Murder weapon is right there. Okay, so we collected evidence from the lab scissors and we processed it for DNA. And our analysis produced a single DNA profile, alright? And that profile, in fact, belongs to someone in this area. Alright? So, how this is going to work, hopefully, okay, is I am going to tell you which shorthanded repeat we're going to look at first. I'm going to put up the allele profile, alright? And we're going to figure out. So everyone needs to stand up for me. Stand up. Everyone's a suspect, staff included. Okay? So, how this is going to work is when I put up the shorthanded repeat, and then I'm going to put up the allele, okay? If you do not match, you sit down. If you match those alleles at that particular STR, you stay standing. You are still in the running. Okay? Oh, yeah, we didn't collect blood, we collected from the
You can send me a narwhal.
That's a good one.
It's Etsy, right?
Alright, so the next piece that I'm gonna put up, right, is blank. You're not gonna, okay? So I'm just showing you which shorthand repeat we're starting with, okay? So when you're looking at your chromatogram, okay, I want you to look at the second window all the way to the right, CSF 1PO, okay? Everyone see where that is?
Mm-hmm.
Alright. If you are not 1112, I need you to sit down. This DNA profile
Here, right here.
is 1112.
You did not know that. Which we already knew, so. You don't kill still 11, 12. Josh,
Wow.
narwhals kill.
That's why you have that big horn on your chest.
Alright.
Not scissors.
So, this is who I have pegged for matching, right? So.
I'm out of the right bed.
I didn't pick those, sorry.
No, but I know I didn't do it.
I would never. Alright. Alright, where are we going to next? We're going third window down, BWA. Everyone see their peaks there? Alright, here we go. 1719. You are not 1719.
Are you still standing, Christine?
Like 17 and
If
19.
you're not 17, 19.
Okay, so I'm only 17, so I said, okay, all right.
She's innocent. Josh is still standing with his stabbing sword. How do you guys know about you?
Of course I didn't do it. Alright, where are we going next? D13, middle panel. You see it? 912. Who's sitting down?
Oh, there are three. There were three.
You had 29. Oh, 4.
DH01. Up top. Ready? 69.3.
So we got 3 in the running still, right?
Great.
Great.
All right. Top. Bottom panel, okay? Let's go. 8-11.
I mean, he is close to the
Oh,
top.
he came back into his own, well, that's good. No, the courts will treat you kinder if you don't try to get away.
All right, so what's the chances of you matching, right? It's 1 in 2.92 billion chances of matching. So, historically that would have been good enough, but today's world population, right? Over 8 billion. So you could in fact have the same individual, which is, if you recall to what I said at the beginning, why systems like FBI have since increased the number of short-handed repeats they rely on to make a definitive match, right? It's up to 20 at this point. And they actually have already proposed further expanding that, okay? But it's because of this, as the world population grows, obviously, the likelihood that you do have a match somewhere, can happen, okay?
The dad jokes weren't good enough.
Oh!
We had our hypothesis, right? Because, you know, beautiful, don't get us wrong. But his unit results, completely different story, right? As in 0, as in not reading anything, okay? So, no, Josh, he was convinced Mills can't do it. Imagine why motive, right? Motive. So rather than coming to terms with his prepping results, he plotted, he planned, and he wanted revenge, and poor Mills suffered. Many a person has been convicted by SDR profiling. None other than Come on, let's baby Mills. Look at him. Alright?
Rest his soul.
Alright.
There's more. Any questions for Allison?
Or Josh.
Or
I was just wondering, you said like there are 8.9 billion people now, but it's quite unlikely that a lot of the ones that would match are in the same country at the same time. So it would still work even if they wouldn't have increased the amount of crime.
Right, I mean, if a crime is committed on a particular day in the
Yeah.
United States and you match a random person, halfway across the world, then yeah, I mean, but I'm just saying that
Yeah.
there is now, because the global population continues to increase, okay, outside of being an identical twin, there is in fact a possibility that you're, based on only 10 STRs, one of which being the next one, that you would match another somewhere in the world.
And of course, lawyers would use that argument, right? But there's a possibility, right? So that's why they've
It
gone
has to
from
be,
20
you know,
into
beyond reasonable doubt, right? And if there's a chance that you match another person, there is, can't be doubt, right? So, yes, so they have increased to 20, short-handed repeats and have already considered further expanding.
In order to do a match, you do need to have fingerprints, DNA fingerprints on file for anybody who could be considered a suspect, right? So
So normally I would have said yes, right? But what's happening now, right, is because so many people will they take it upon themselves. They take, you know, at-home DNA tests for diseases and for hereditary things, right? And then people will put their DNA sequences in different databases on their own. And so what happens is you don't necessarily match to the perpetrator, but because these things are passed down from family, you can see familial relationships by comparing your evidence to public domains like that, or public databases, right? And so it would be enough probably to get a warrant to sample family members if the evidence you collected at crime scene happens to match with a high enough frequency to one of those publicly available DNA sequences. Does that make sense?
And it's an advantage, going up to larger numbers of STRs, because then with greater precision, you can identify, if there was a family member related to the individual who perpetrated the crime. You can figure out those relationships if you've got more, more data, more STRs. Oh, the Golden Gate Killer in
Yes.
California was
Right, that's
identified that way, right?
how they were able to catch him, actually.
Like 40 years later, when I was a kid, growing up in Northern California, it was a big thing.
Yeah,
And
back
then,
in the 70s,
yeah, and then only recently identified, by DNA analysis. So that was a big story, and there have been other ones like that.
But when you give something to 23andMe, you're not giving they're not doing your entire genome, right? They're looking for very specific markers.
They're looking at STRs and some other markers.
Oh, they are looking at STRs. Oh,
And some other markers. Yeah. But they're not sequencing your whole genome for $100.
Right.
Whatever.
It's incredible that prior sites to find new STRs, have to apply to the whole human population.
So I think I understand what you're asking. So the primer sites that the 20 primers bind to are highly conserved sites. The STRs in between are not so conserved, but where the primers hybridized have to be very conserved in the human population. Yeah.
Yeah, that's how they're designed on either side of a particular STR, but they have to be designed based on a conserved region, so that, yes, they would amplify in all populations.
Sorry, so that's how the gene test, is that how they establish that you have like a second cousin living wherever, and they establish based on the same STRs?
We're going to do that in just a second.
Any other questions?
What about the touch DNA? How accurate is the touch DNA?
So touch DNA, if I touch the desk here with the finger, I've left a fingerprint there, but I've also left DNA there from skin cells. And what they do is they can lift those skin cells using different techniques, isolate the DNA, do a profile. So touch DNA is just, it's like, your mouth swab, or if you were chewing gum and you throw your gum away or whatever. It just means that it's from a surface that somebody touched. And surfaces like this, clothing, they can get DNA off of almost anything these days.
Any other questions?
Okay, so we have another piece of information we can get from our data, and that is some of you have been asking some very nosy questions about about how related the staff are. I'll tell you up front right now that amongst the staff there are 4 familial relationships. 4. I'm going to put the data up on the board, and you're going to tell me what the familial relationships are. So, the first one I'm going to put up is, and I have to say, there are 9 informative loci here. The 9 STRs, the amelogenin one, there's only 2 variants, so we're not going to use that one. We're just going to use the 9. And each of us has 18 different data points then, because each of the 9, there's each chromosome, right? So if you're an 11-12 heterozygote, that's 2 data points. If you're an 11-11 homozygote, that's 2 data points. So we have 18 alleles we can look at. And so the relationships are as follows. Mills and Kalani, they share 5 out of 18. Allison and Steve share 5 out of 18. Allison and Katie share 8 out of 18. Allison and Kalani, I'm going to warn you ahead of time that these relationships may not be what you thought. Allison and Kalani, 4 out of 18. Nils and Katie, Nils and Katie, 5 out of 18. Nils and Steve, Steve, 6 out of 18. Katie and Kalani, Katie and Kalani, 8 out of 18. Steve and Katie, Stephen, Katie, 8 out of 18. Nils and Allison, 4 out of 18. Steve and Kalani, 10 out of 18. That's all. Okay, so the data here can help us with the complex familial relationships we have on the staff. No inbreeding, I'll tell you that. So I'll tell you that if you look at the data, you should be able to make some guesses. And this would be based on not only the data here, but taking into consideration the age of the staff members. So what's the obvious one here? Okay, so what does that suggest? 10 out of 18, just a little better than 50%, right? So what might that suggest? Anybody? Familial relationship?
Father-daughter.
Father-daughter.
Yeah. Is that you? Searching for it. I've always
Oh, dude,
You might
remember?
have.
So, yeah.
I always wondered.
Is anybody else related up here? Steve, Katie, what do you think that one is? Any guesses? Almost 50%. Not so easy, is it? You could also go by, not to say age, but appearance, right? Well, let's leave that one for a second. What about Katie Kalani? Cousins. Good. First cousin. Okay, so what does that tell you about this one then?
Uncle?
What about Allison and Katie? Cousins. Yeah, could be. Cousins? Sisters? Katie and Allison have
That's a good one,
similar
too.
accents, so we didn't
Yeah.
take that from the same part of the
Cousins.
country. So this is actually a warning from, this kind of data, right? By random chance, you can end up having similar profiles. So if we looked at all your profiles in the room, we'd see that some might have a 3 out of 18 match, and some randomly might have an 8 out of 18 match. So this can happen randomly, and in fact, in this case, there is no familial relationship there, okay? So, there are 3 familial relationships up here, and I told you there were 4. What's the 4th one? Mills and Allison. I cheated on that one. But it's really good, their fingerprints are really different. All right, so that's what we can glean from the fingerprint data. Now, if you look at fingerprints like Kalani's and my fingerprint, it's more than just a 10 out of 18 match. For every single locus, for every single one of those 10 loci, one of my alleles has to match one of hers. So it's even more than the 8 out of 8 or the 10 out of 18. There's one allele that has to match for every single locus. So that's another piece of information that you can get. That's not true for cousins or uncle niece, but for father-daughter or mother-daughter, half of the alleles have to come from one parent and half from the other. So that's an additional piece of information. And in paternity cases, this kind of thing is used all the time, this fingerprint. If you are, in fact, the father of a child, then half of your alleles are going to match between those 2 individuals. And if you're not the father, then it's not going to be like that, especially if you're looking at funny loci. Okay? So, there you have it. Now you know the whole story. All right. Any questions about this? Or about the fingerprinting from
Oh,
before?
it's sad, this is gonna go away
John
when
is
he's
still
done.
sitting here, that's good. Anyway, we know where you live, John. Can't get away, we know where you live, we know where you work. Okay, so we're done with fingerprinting. We will take a 5 to 10 minute break now.